Okay, we're going to go ahead and start now. Hopefully everybody can hear me. I know you all can. So I think I've got the speaker thing working for us. So we're going to wrap up the chapter on chemistry today. You know, control your enthusiasm on that. And if we have time, we'll go ahead and start cells. Now, the lecture test for this will not open. I'm not going to do anything about the lecture test till sometime next week, because we've got the lab test to go through and get that done. And I'm going to leave that open. That'll open up for the online, for the people that are taking it online. It won't open up until I give out yours today at 1 o'clock. Whoever's coming here today to take it. So, and I'll tell you, it's pretty straightforward. I've rewritten it 3 times to try and keep it as okay. So to keep it as unambiguous as possible and use the best possible images. The way we're going to do it in class is that I'll have the images on the PowerPoint and the test will have the question. And so all you gotta do is look at the PowerPoint. It'll be real obvious when I always hate saying something like that, but it'll be real obvious where there's a question, there's got to be a picture to it also. And plus the picture will be numbered, so you shouldn't get confused, unless I messed up on the number, which is entirely possible. Anyway, there is no Zoom lab this afternoon, so don't plan on coming into lab via Zoom, because we're not meeting, because all we're doing on the ground is having the test and then getting out of here. And the Zoom test, the Zoom lab, you don't have to Zoom this afternoon. That's what I'm trying to say. So, okay, before I confuse myself any further, let's get into chemistry and get done with it. Anybody have any questions, by the way? You're just so excited to get into chemistry, you can't wait, right? I like sucking up, it's good. Yeah. The test if you are taking it online, it'll be an e-learn. It'll show up today at 1 o'clock. It will say AP1 Lab Test 1 Spring 2022 or something like that. And you just click on it and follow the instructions. You will not need to use the lockdown browser or anything to take this first one. So many of you are not familiar with it. I don't know if you are familiar with the lockdown browser or not. Sometimes it works, sometimes it doesn't. So on this first test, first lab test, I'm not going to use it. So, okay. So you should be able to find it online. And for those of you that are going to take it, in Zoom, or you don't have to be in Zoom to do it. Just if you go into eLearn, go to under assessments, I think it's called, and then click on quizzes, and it'll pop up there as test number 1. Okay. Any other questions or comments on anything? Okay. Now let's get started here on proteins. We've already talked about carbohydrates. We've talked about lipids. We wrapped up lipids last week with the prostaglandins. And now we're going to go into the third category. Here we have the proteins, and the proteins are a pretty fair amount of our body. 30% of our cell mass. Proteins are involved in structure and in chemical reactions too. Proteins make up all the enzymes. The enzymes are the biological catalysts that enable reactions to occur at body temperature, at low concentrations and small particle sizes. Structurally, all of our skeletal muscle are protein. All of our involuntary smooth muscle is protein, and so is the cardiac muscle. The We have protein that wraps around our bones to make them stronger and more resilient to stress. That's collagen. We have collagen fibers that form the tendons and ligaments in our bodies that hold bones and muscles together. So, not to mention all the other chemistry aspects of it, not just enzymes, but antibodies for fighting infection are proteins. So there's hormones are proteins. Most of our hormones, the things like thyroid hormone and growth hormone and things like that, are all based on proteins. So if you are a protein molecule, you are held together, made up of amino acids linked together with what are called peptide bonds. It's a type of chemical bonding they have. You are usually going to have carbon, hydrogen, and oxygen, as well as nitrogen, because amino acids have what we call an ammonia group or an amine group attached to them. That's why they're called amino acids. Occasionally, we'll throw in a sulfur and a phosphorus in there for an additional type of bonding. But if you're a protein, you're going to always have carbon, hydrogen, oxygen, and nitrogen. There are 20 different types of amino acids. That's all, more or less. That our bodies use only 20 amino acids, and we can link those amino acids together in any order, in any sequence, at any length that we want. Some protein chains are 300 and 400 amino acids in length, and so obviously they're going to have to repeat the use of various amino acids again and again and again. And they do. Hemoglobin, for example, is the protein that carries oxygen in our blood. It's 360 amino acids long, so it's a fairly long protein. Obviously, with only 20 amino acids, we are rearranging the order, and we're putting amino acids, repeating them over and over again. But if you consider there are only 26 letters in the English alphabet, but the average dictionary contains about 200,000 words, it's fairly easy to understand that all of our proteins are made out of these 20 simple amino acids. An amino acid always has the same structure. It always has an ammonia group on one side of it and an acid group on the other side, and it has a special, what we call an R group in the middle. And those little R groups are a part of the molecule for the amino acid. That specialized R group determines what the amino acid is going to be able to do. And it's very important that we put the right amino acid in to our protein. We call it a protein chain of amino acids, and it's all controlled by our DNA, and mistakes happen sometimes in making these proteins. For example, in hemoglobin, the number 6 amino acid out of 360, the number 6 amino acid is called glutamic acid. We'll talk a lot more about that later on, but it's glutamic acid. However, if you substitute valine in place of glutamic acid in the number 6 spot, then your hemoglobin becomes sickle cell hemoglobin. And that doesn't happen randomly. That's genetically controlled because our DNA controls the production of all of our proteins. But sickle cell disease is called the physiological cause of sickle cell disease is putting valine in place of glutamic acid, and they are very, very similar in their structure. One little mistake like that has extremely significant consequences in an individual's life. So the order of these amino acids is dictated by our DNA, and it's very important that they follow that particular order. The sickle cell DNA, sickle cell hemoglobin is following the instructions of the DNA, which has the mutation in there that's allowing the wrong amino acid to go in. Anyway, this is what an amino acid looks like. You've got the acid group on one end, that's what we call a carboxyl group, It has a carbon atom and double bonded to an oxygen and another hydroxyl, an OH group there on one end, and you have the ammonia group. You can see it's nitrogen and a couple of hydrogens on the other end. And in the middle is this R group, the side chain, that dictates what the amino acid is going to be. Some we may have heard, and I've mentioned two of glutamic acid and valine. If you've ever used equal, you all know what equal is, right? Aspartame, the stuff that comes in the blue packets, at least it used to anyway, artificial sweetener, that is basically an amino acid called phenylalanine. And most people that use it for making coffee and tea, drinks, sweet. Aspartame may often also be used in various low calorie or diet soft drinks. At least it used to be. I don't know if it is anymore, but it is basically an amino acid. It's called phenylalanine. So, and if we drink a lot of, if we use a lot of equal, we're getting a lot of phenylalanine thrown into our diet. So, anyway. Peptide bonds are a type of covalent bonding that occurs between the amino acids. They link together. They link together in whatever chain that they're supposed to be when they're being assembled. The proteins are made inside our cells under the direction of our DNA, and we have all these amino acids available to us from our diet. We eat protein and we break that protein down, and whether it's animal protein or plant protein, we tear that apart and take those amino acids and reassemble them in the right order. Because glutamic acid from a plant, from a vegetable, is the same glutamic acid that comes from a hamburger, if you will. So, let's see if this works.
Proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
I like how the video stops and just leaves you hanging right there. Like, oh, I got to know now. Basically, when a cell makes a protein, these little purple beads here represent each of the amino acids, and whatever the order dictated by the DNA is the order that the amino acids are going to be linked one to another. And so we have this first, we have the cell will produce a long chain like this with every amino acid supposedly, hopefully, in its right spot in here. Now, our cells don't leave it like this, because if you have a long strand of protein, it's likely to get broken. It's sort of why our chromosomes condense before they undergo mitosis, because the DNA strands that we have in our cells in the nucleus are 6 feet long, and they're microscopically thin. And so, if you're going to be dragging one of those long strands across from one side of the cell to the other, the odds of it breaking apart are pretty good. But if we condense it and get it real thick, they don't. Well, the same with the proteins. We don't leave it in this long chain structure like this. We will.
Most proteins.
We don't want to watch you again. We didn't like you the first time. Okay. So the primary structure of the protein is just a long chain. When the cell produces the protein, it comes out of the cell in this long chain, but they don't stay that way. They're going to be folded and twisted and bent and curled into one of 3 different shapes. So we had the primary structure, just the long chain. The secondary structure is we're going to curl or change the shape of the protein to fit its job. Because many proteins work in our bodies by landing on very specific, what we call active sites. And for example, if you are a hormone based on a protein, you're going to land on set. You're going to work on cells that have a site that will receive you. It's sort of like a lock and a key. We shape the protein in the form almost like a key, and it will only land on sites where it has a lock that fits it. Because, we all know with locks and keys, you have to have the right lock to go into for the right key for the right lock. Well, this is the same way our bodies function. Proteins only work on cells that have a place for them to land. For example, our thyroid controls all of our metabolic activity in all of our cells by releasing thyroid hormone. So every cell in our bodies has a receptor site for thyroid hormone, which is a protein. And that protein can land on every cell. However, our brain controls the release, controls the activity of producing thyroid hormone by releasing something called thyroid stimulating hormone. Now, where do you think thyroid stimulating hormone works? On the thyroid, okay? But it will circulate through every cell in our bodies. It will go around every cell because the brain can't say, go to the thyroid and nowhere else. Thyroid stimulating hormone will circulate in our plasma to every cell, but it will only work in the thyroid because the thyroid is the only one that has receptor sites. The receptor sites are proteins, and the hormone is a protein, and they will bond together to do their job. It won't work anywhere else. So we shape these proteins around to do things for us. Secondary structure, the alpha helix, very common type of shape here for our protein. It sort of resembles how a DNA strand looks. The alpha helix looks something like this. And so elastic fibers are like this. The alpha helix, shaped like a spring, can expand and contract. The elastic fibers in our skin doesn't tear when we do things. When we stretch out our arms and legs, the skin doesn't tear. Our muscles are elastic. They're springy and can contract and relax. Our blood vessels have elastic proteins around them so our arteries can expand when a surge of blood goes through and then snap back when it's passed. We call that the pulse. We can detect our pulse as blood goes through our arteries. So these elastic fibers, our hair is full of alpha helix proteins because our hair can be bent back into any shape we want or don't want. When you wake up in the morning and you've got a bad case of bedhead, a little work puts it back to reasonably where you want it to be. Because it can change its shape and then be put back with no apparent damage. That's what the alpha helix lets you have. The pleated sheet, which looks something, like a staircase, if you will, gives you tremendous strength. Your collagen fibers are held together in a pleated sheet structure. Collagen fibers, of course, are the most abundant protein in the body and are individually stronger than steel. I say that every time, but it's true. The collagen fibers that make up, for example, our tendons are lined up side by side in almost a straight line structure. And so they have great strength because they're lined up like this. And this pleated sheet is how they're organized so that they have these folds built into it, but the protein strands are all one after another next to each other. It's that dense connective tissue we talk about in lab, stuff that makes up like our Achilles tendon, for example, is a very, very powerful structure made up of collagen, usually in some sort of pleated sheet structure. Spider webs, by the way, totally unrelated to us, but spider webs are built with protein and it's a pleated sheet structure. Many spider webs can survive high winds and rain and storm events outside all the time, particularly in the fall when we see those big old black and yellow orb spiders that have they're about that big, and you think they're going to kill you, and they're going to build a web that's literally about this far across, and it will survive all sorts of events that you expect it to get blown away, but it's there because the protein that makes up the spider web is so powerful, and it's all in this pleated sheet structure. And you have to be very careful how you say pleated sheet so you don't say it too quickly and say the wrong thing. Anyway, third structure is the tertiary structure. The alpha helix and the pleated sheet structure are often combined in the same strand of protein, depending on what it's doing, depending on how you want to configure the protein, because we can cause the protein to bend and twist and form whatever shape we want. And so the tertiary structure usually has both the alpha and the beta components built into it. And then the quaternary structure is simply a collection of the tertiaries. The more complex the protein is, the more likely is you're going to see these tertiary structures built into it. For example, hemoglobin has 4 tertiary structures put together in its overall shape. It's shaped sort of like a cube, if you will, with with 4 different subunits. And let's see, there we go. There's at the very top of the slide, you see the primary structure, just the straight chain approach. Then you can see the pleated sheet. That's better than my pleated sheet, but I like my helix better. But the helix looks like, if you want to know what the helix looks like, think of a telephone cord. Telephone cord is a helix. It's a spiral helix, and that's what the alpha helix looks like. The pleated sheet, they say it looks like it's folded like an accordion. Who looks at accordions? These days. But anyway, it's folded. I think it looks more like a staircase. That we have these, but the fibers are running side by side. The tertiary structure is made up of both the alpha and the beta secondary structures, and then there's your hemoglobin, for example, as your as we call it, the quaternary structure. Now, if you're a protein, you come in 2 ways. You're either fibrous, which is usually structural, or globular, which is squishy. The fibrous proteins, if you eat beef or chicken or fish or any kind of vegetable protein, you are usually consuming fibrous proteins. They're shaped like strands of, you can see the strands of fibers linked together in beef. You can see the grain of the beef, or in the chicken. You can see when you pull the chicken apart or the fish apart, you can see the strands in there. They don't break down when you cook the food, whatever you're cooking. These types of structural structural proteins keep their shape regardless. They're not affected by temperature changes or pH changes. They give us mechanical strength and what we call tensile strength. Mechanical strength and support, they hold our bones together, so we have a distinct shape to our bodies. Tensile strength, they are very resistant to being twisted and compressed. One of the reasons our bones are so powerful is that they're wrapped layer by layer with collagen. And so our bones can resist twisting a great deal. Keratin, the protein that waterproofs our skin, is a fibrous protein. Elastin is a fibrous protein. Collagen is a fibrous protein. The contractile fibers in skeletal muscle and smooth muscle and cardiac muscle, the actins and the myosins, are all fibrous proteins, and they will survive even after death. It takes a long time for the fibrous proteins to degrade and to decompose, whereas the globular proteins, the other type, the squishy ones, are gone almost instantly. But we need to have both types. The globular proteins are the functional proteins. These are the antibodies that we produce in response to a foreign antigen. A foreign antigen is a marker on a cell that identifies it as self, but it may not be our self. An antigen is if you get strep throat is caused by a bacteria, what we call a streptococcal bacteria, and the bacteria have these little markers on the surface of their cells saying, "Hey, I'm strep type A," and the other one's like, "I'm strep type B." But they have a marker that says what they are. And our body recognizes that marker as foreign. And those foreign markers are called antigens. All of the cells in our bodies have antigens to identify us as self, so our own immune system doesn't attack ourselves. When we do attack these foreign antigens, though, we produce antibodies to glom onto these foreign substances and destroy them. We also, most of our hormones are protein-based. And again, they're very squishy, large collections of molecules together. There's something here called a molecular chaperone. Molecular chaperones actually function like a blender. They'll take a straight chain protein and twist it around to assume the shape it needs to be an enzyme, to be a alpha helix or a pleated sheet or a quaternary protein. They have the directions on how to, a straight chain protein comes in and out comes the quaternary structure of hemoglobin. Here comes a 360 chain of amino acids to be hemoglobin. It's not going to work until it is twisted into that right shape. That's what the chaperone does. And then the enzyme, the biological catalyst will cause chemical reactions to occur at a much faster rate. And because, again, we can't control temperature, we can't control particle size, and we can't control concentration. But we can make reactions work by using enzymes. Problem with proteins. These globular proteins are very sensitive to being denatured. Denatured means that they are broken apart by changes in temperature or in pH. If we get too hot or too cold, the shape of the globular protein can change, and that's going to affect its ability to function. The proteins have what are known as active sites where they will bond to a cell. And if the active site's broken, then the protein can't function anymore. A change in temperature, either up or down, a change in pH, either more acidic or more basic, the protein becomes denatured and becomes unusable. And if it's allowed to continue, the protein is destroyed. It becomes irreversible. Now consider an egg. We've all seen eggs. We've all eaten eggs, I think. The yolk of the egg is all cholesterol. The white of the egg is albumin. Albumin is a very common protein in our bodies, but it's also the white of the egg. Now, when the egg first comes out of the shell, the albumin is clear. It's more like a gel, but it's still clear. When you cook that egg, the albumin will solidify and turn white. We call it the white of the egg. This is not a cooking lesson, but this is what happens. Now, the albumin has become denatured. We can't reverse that. We can't take the egg and reverse the process of cooking it so that the albumin goes back to its semi-liquid state again. It's a permanent change. It's been denatured. It's gone past the point of where it can recover from the temperature change. Our bodies are the same way. If our bodies get too hot, proteins start breaking down. If they get too cold, they start breaking down. If we can alter that temperature change or a change in pH, the proteins can recover. If it becomes too extreme, then the cells start dying, and we may too. So, okay. Now, of all these different types of proteins, probably the most important one we pay attention to are the enzymes. The enzymes are a biological catalyst. And a catalyst is a substance that makes chemical reactions go faster. It's like putting lighter fluid on charcoal. You could throw, I think I said this before, you throw a match at a pile of charcoal and you can throw matches at it all day and nothing's going to happen because it's, well, you know, the match will go out and the charcoal won't start to burn. But if you soak it with lighter fluid, the lighter fluid will help get the charcoal to burn at a faster rate and it becomes self-sustaining. That's what a catalyst does. Biological catalysts work for us by allowing us to not have to worry about the small concentrations, the fixed temperature, or the small particle size. A catalyst will speed up the reaction in our bodies and will not be damaged on its own. It lowers the amount of energy. Now, how does this work? Well, down at the bottom of the picture there, you've got that enzyme, and the enzyme is just a protein. And the enzyme has what we call active sites on it. You know, the enzyme has active sites on it, and it will cause a chemical reaction to occur. Now, normally, if we were going to mix together baking soda and vinegar, we've all done this or seen this at some point. Baking soda and vinegar, you mix it together, what's it do? It foams and bubbles, and you get the carbon dioxide. And you, when we're kids and we do it, we get a lot of bubbles. That's the carbon dioxide. We also get water and salt. We're probably not even paying attention to that because why would we? But we get, so that kind of chemical reaction gives us the release of carbon dioxide, baking soda, salt, and water. And we mix baking soda and acetic acid together. We always mix it together in a container and it bubbles out. It does just fine. Well, now suppose you had a beaker, a large beaker filled with water, and you added 1 molecule of baking soda and 1 molecule of vinegar. Vinegar is acetic acid, so that's the acid, and baking soda is sodium bicarbonate, which is the base. And when they come together, they're going to react and release carbon dioxide and give you salt and water. Well, you've got a 10-liter beaker. I've actually, they do make them that big. I don't know why they have them, but they're huge. So you have 1 molecule of baking soda and 1 molecule of vinegar, and they're just randomly floating around in 10 liters of water. When do you think they're ever going to come together? Extremely unlikely, right? Because you have these 2 little molecules that sort of, depending on the movement of the water molecules. Nothing we can do it. We can't. That's what it's like in our body. Because the very small concentrations of substances are depending on the movement, you know, the passive movement of water or air for a reaction to occur. But suppose we could add an enzyme to that beaker. Suppose we could add an enzyme to the beaker that would grab the baking soda molecule and grab the vinegar molecule and bring them together and do it very rapidly. That's what an enzyme does. The enzyme will grab the reactants and bring them together. Imagine that if this classroom was completely empty and we had 2 volunteers that were from opposite sides of the room. So you're at one corner and you're in another corner, and you're blindfolded and you have earplugs in. There's no other furniture here. It's wide open, and you're told to go out and randomly move through the classroom until you can shake hands with her. And she's trying to find you to shake hands. Okay. So there you are, just groping. You can't hear him. You can't see him. You're groping to find the other person to shake hands. You might be here all day, all week, who knows? But then the rest of us come in. And immediately you go over and grab her hand and you grab his hand and you bring them together. You shake hands. And then you let go. And then you come over and grab him and you grab her and do it again and do it again and do it again. So the enzyme, all it does is bring the reactants together instead of waiting around for them to randomly collide, which they may eventually, because that's how chemical reactions usually occur, is the random collision of molecules. The enzyme brings it together. The enzyme takes the substrates, that's the reactants, and brings it together so they can go at a much faster rate. Instead of waiting for it to randomly occur, the one thing we can do is we can make lots of enzymes for whatever we need. And we have all sorts of enzymes in our bodies. We can allow chemical reactions to occur to build up molecules and to tear it down. In our mouths, we have an enzyme called amylase. We actually start the process of digestion in our mouth with breaking down carbohydrates. When we eat starchy foods, the enzyme amylase starts tearing apart the starch to convert it to glucose. And so the amylase is, the amylase this protein chain has a spot on it, an active site on it that only starch can land on. And everything's going to try to land on it, but it's not. Only starch can land there. When starch lands there, then that causes the reaction to occur that pulls it apart. And that's the only place that amylase can work. It only works in our mouth. And so we end up with a product here. So we have, on every enzyme, we're going to have active sites and we're going to have substrates. The substrates are the reactants. Now, different types of enzymes. We have two parts usually to an enzyme. We have what's called the apoenzyme, which is the protein portion, and a cofactor. It turns out that we use vitamins as the cofactor or the coenzyme. It's not an enzyme. The cofactor or the coenzyme is not an enzyme. It just helps the actual protein do its job. Vitamins help us a lot in this. Enzymes are very, very specific. They are designed for one substrate, one set of reactants and only one set of reactants. Amylase only works on starch. It doesn't do a thing on fats or proteins. So they are very, very special. Anytime you see the ASE ending on a word, that means it's an enzyme, like oxidase. Oxidase uses oxygen to complete a reaction. Hydrolase usually pulls water out of a substance as part of its reaction. So these enzymes are very, very unique, and we've got millions of enzymes we can draw on. Classic answer for what's an enzyme do? It lowers the activation energy. Because it takes, if you have two molecules bouncing around until they run together, that takes a lot of energy. Now, they're not doing anything, but they're getting pushed around by the air molecules or the water molecules that they're in. And that takes a lot of energy for that to happen. If we were to apply heat to that system, the molecules would randomly combine at a much quicker pace, and that would be fine, except we're not allowed to heat people up to get things to happen like that. So we can't do anything we can't apply energy to get the reaction to go, but we can apply enzymes. So now, and this is, oops, let me, here.
And it's a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
there they go, leaving us hanging again. It's like, and where is it? What's the rest of it? Well, but essentially that's what they do. All the chemical reactions in our bodies are controlled by enzymes because we're stuck at 98.6. We're stuck with small particle sizes and small concentrations. Here we see an enzyme linking together amino acids. The active site is where the enzymes can bind to the amino acids. The amino acids, in this case, the reactants, are always called the substrates. They come together, they have a bond that forms, and then they get released, and the enzyme goes back out and does it again. That's the beauty of the enzyme. It doesn't get changed as a result of the reaction.
The enzyme doesn't change?
The enzyme does not get changed. It brings the reactants together. The reaction occurs, whether it is to tear it apart or to build it up, and then they let it go, and it doesn't matter. The enzyme is ready to go do it again. Now, problem here, enzymes are very susceptible to being denatured, just like any other protein. And when the protein gets denatured, it breaks bonds in the structure of the chain. And so in this case here, we're looking at an enzyme. Normally what will happen is that the substrates will land on the active site of the enzyme. The shape of the substrate is dictated by the shape of the active site, but there they land there, they're happy, I guess, and they do whatever they're going to do, and they either break apart or combine. But suppose we heat up the enzyme, the active sites, or cool it down. The active sites, the shape of those active sites where the reactants would land are altered, and they don't fit correctly anymore. The reactants haven't been changed, but the protein has. And so the enzyme is no longer able to do its job. And so it can't. The reactants are not going to come together. They're not going to fit in the active sites. Yeah.
This is before the reaction occurs.
Exactly. You'll never get, when the enzyme becomes denatured, you don't get a reaction at all. They're not going to fit there because something is changing it. Now, whether it is a change in pH or a change in temperature, when an individual goes into cardiac arrest and they're no longer removing carbon dioxide out of their body, what will happen is the cells take a long time to die. Muscle cells take up to 12 hours to die after the heart stops. Brain cells shut down in 5 minutes. Muscle cells will continue to burn glucose and release carbon dioxide for hours. That carbon dioxide goes into our blood and is converted to carbonic acid, making us more and more and more acidic. And that's going to destroy the enzymes in the cells. So how do we respond to that? Well, in addition to trying to get the heart started again, one of the therapies is to administer large doses of sodium bicarbonate, baking soda solution, to the patient to neutralize the acid that's forming in their plasma. Because the cells don't know the heart stopped. Cells don't know they're not breathing. All they know is, hey, I got some glucose and I got some stored oxygen. I'm going to burn some glucose and release some carbon dioxide. We have to counteract that, because when the heart stops, it isn't like everything shuts down immediately. It takes a long time for muscle cells, and the bulk of our temperature and the bulk of our carbon dioxide is usually coming from our skeletal muscle cells. So yeah, the change would occur before the reaction could even start. So there's a long answer to that, but a short question. Okay, so that is protein. And we all, we get our protein in a variety of ways. There's plant proteins and animal proteins. It doesn't matter what kind of, what our food source is for proteins, because they're both plant and animal proteins are going to have the same amino acids. As long as we get all 20, we're just fine. Now, the last category of organic molecules are the nucleic acids, the DNAs and the RNAs. These are always going to have nitrogen and phosphorus in addition to carbon and hydrogen and oxygen. The DNA strand, typically each strand of DNA is about 6 feet in length, a little bit less than 2 meters. And the basic unit of these DNAs and RNAs, these nucleic acids, are the monomers are called nucleotides. And a nucleotide will consist of a nitrogen base. It will consist of a 5-carbon sugar, either a ribose or deoxyribose sugar. They're both 5 carbons, and a phosphate group. Now, the phosphate and sugars tend to make up the backbone of the DNA molecule and the RNA molecule. It is the bases, the nitrogen-containing base, that determines our genetic code. It's the order of these bases that determines our genetic code. So we have DNA and RNA. They're all made up of nucleotides. It's a terrible picture here. Very little difference between the structure of the sugar in the DNA and the RNA. One is a ribose sugar, one is a deoxyribose sugar, which means that there's in the DNA, we're missing 1 oxygen. That's all. They're both 5-carbon sugars. Just a little bit of difference in their structural arrangement. You have a sugar and you have a phosphate group and you have a base, and it looks something. No, we don't see her. Okay, so help yourself. Do you want some of the little ones?
No, this one, sorry to interrupt.
That's okay.
But yeah, they're like panicking on those tests.
Mm-hmm.
We never do that.
No, everybody's getting a test today. There's testing chemistry. Okay, our DNA is our genetic makeup. It is the basis of what we are. The who we are depends on, experiences and knowledge and skills, all the other stuff, all the sort of the touchy-feely things. What we are is determined by the DNA. It controls the entire makeup of all of our proteins. And we are mostly protein in one form or another. And the DNA strand is a double helix. It's shaped like if you imagine a ladder that you could twist so that it would look so you have your DNA strand essentially looks like this, but imagine that strand is twisted. It's not too bad, like that. So you're if you lay the DNA out flat, it would look like the ladder on the left, but it really is a double helix, like a double spiral staircase. Now, the nucleotides in DNA are going to contain a deoxyribose 5-carbon sugar, a phosphate group, and a nitrogen base. And there are only 4 nitrogen bases: adenine, guanine, cytosine, and thymine. And these bases will pair up with each other in that ladder. So, for example, the way the nucleotide is arrayed, the nitrogen bases are always in the middle of the ladder. And so you would have adenine pairing up with thymine, guanine pairing up with cytosine. So this is what we call complementary base pairing. Adenine always pairs with thymine. Guanine always pairs with cytosine. The ladder-shaped structure here, let me get this. So we got a better picture of this than what I'm trying to draw. Here we go. There's your double helix. There's your double helix right there. Now, the adenine, in this case, if the adenine is represented by the red bar, that is the nitrogenous base adenine. it will always pair to thymine. Anything else is a mistake. Now, do mistakes happen? Oh yeah, mistakes happen all the time in our DNA. But adenine will always pair to thymine. Guanine always pairs to cytosine. So here we see adenine in red and adenine here and adenine here and adenine here and here and here. And it doesn't matter which side of the strand it's on. If there's adenine on one side of the double helix, there'll be a thymine on it pairs to. And the order of these bases dictates the order of the amino acids, that how we assemble the amino acids in a protein. So every protein our body makes is controlled by the DNA strand by simply having a unique sequence of bases. So, and you'll see that there are no errors on this one. Here we see cytosine green pairing to guanine. And guanine, there's guanine there, and there's cytosine there, and there's guanine there, and there's cytosine there. So adenine, and generally, this is how it should pair together. The adenines always pair to the thymines. Doesn't matter which side of the strand. The guanines pair to the cytosines. Doesn't matter which side of the strand. The order of these bases is very, very specific. The order of the bases dictate the order of the amino acids that we have to assemble to make a protein. The DNA is a cookbook for us. How to assemble a human is all contained in our DNA. Now, can you make a copy of somebody? Well, we clone animals. We clone sheep and cows and horses and clone lots of plants. But is that going to be the exact same person? No, of course not. Physically, they may be, but psychologically, emotionally, intellectually, they're going to be completely different. We don't know what it's going to produce. They just look the same. That's all. So we can't duplicate the life experiences. We can make another copy of it. And somewhere in the world, somebody has probably cloned humans. It's generally it's against the law in most countries to do that, but someone probably has somewhere or has experimented with it. For whatever reason, I don't know. Sometimes just because you can do it is not a good reason to do something. It's one of those hold my beer moments, type of things. So I'm going to go copy some people, Anyway, now we're going to talk a lot more about how the DNA strand functions and how we put things together when we get into chapter 3. But it is pretty remarkable that the error rate in our DNA, it's okay. And we're all going to stare. Our organist at church has that same ringtone, and she's hard of hearing, and so it'll go off in the service during the sermon usually, and she'll just let it ring and ring, and finally she'll realize, oh, that's me, and then she'll turn it off then. So it happens. So anyway, okay, so now to make DNA work, the DNA always stays in the nucleus of all of our cells. Every cell in our bodies has the exact same DNA. Whether it's a skin cell or a liver cell or a nerve cell or a muscle cell or a white blood cell, it doesn't matter. The DNA is all there. It's all the same DNA. It's all whatever makes that a unique cell, like a liver cell or a skin cell or a bone cell or whatever. That's what's turned on. Everything else is turned off. Everything else is there, but not being used. And so the DNA never leaves the nucleus of our cells. It gets this other nucleic acid called RNA to do its work for it. if we want to make a protein, let's say we need some more hemoglobin, or we need some more hair color, or eye color, or pigmentation, some more melanin, or something like that. The DNA will instruct the assembly of an RNA strand to occur. Now, the RNA is not a double strand, it's only a single strand, and it makes a copy of the section of the DNA. Now, you've all heard the term genes, right? Okay. A gene is nothing more than the physical location on the DNA strand that contains the information to make a protein. Hair color, blood type, skin color, body size, dangling earlobes or not, dangling or attached. Some of the more ridiculous, not ridiculous, but unusual, how about unique types of traits? You know, can you curl your tongue? If you can curl your tongue, that's a dominant trait. Yeah, can you curl your tongue? You know,
Yeah.
Okay, there you go. And those of us that are normal can't, but you all got that pretty well too. Sometimes I had to, some classes over the years I had to explain that. You know, tongue curling is a dominant trait. I don't know why, but so is blood typing is okay. You were sucking up earlier, now you're just showing off. So blood type, hair color, eye color, things we inherit from our parents, dominant and recessive traits.
Widow's peak.
Widow, oh, that's a good one. Widow's peak, yes, is a recessive trait inherited in males from their mothers. You know, so if your father or your brother or a male husband, male friend, whatever, has widow's peak, he can thank his mother. He can also thank his mother if he develops male pattern baldness. So my wife always says I have male pattern blindness because I can open the refrigerator and say, "There's nothing in here. You know, it's not here." And then she'll go over, "There it is." You know. So anyway, if we need to produce hemoglobin or any of the other proteins, we will form a strand of RNA right at the site of the gene. The gene is the physical location of the instructions, and the RNA then will move out into the cell from the nucleus and direct the assembly of the amino acids. And then when it's done, it'll break apart, ready to go do it again for something else. So the RNA is made up of nucleotides, the bases. It's going to have the phosphate and the sugar and the base. The phosphate and the sugar are the backbone of the strand. This is not a double strand, it's a single strand, and it's going to have almost all the same bases. It has it uses uracil in place of thiamine, and don't worry about that right now. And it comes in 3 varieties. We have what's known as messenger RNA, which takes the actual instructions from the DNA out to the cell. We have transfer RNA, which actually physically brings the amino acids together to link them into the protein. And then we have something called ribosomal RNA, and they produce the site of the assembly on what we call the ribosomes. And we'll get on to that in chapter 3, which is coming up next. So we can make 3 different types here, and the whole thing comes together. And then as soon as the protein gets made, it all breaks apart, and all the nucleotides are put in reserve, ready to go again. And it just happens over and over and over again, because we're always making proteins because they're always wearing out. And so we use these 2 nucleic acids. The DNA is the DNA is the cookbook, and the RNA is a copy of a recipe that Grandma has this cookbook with all of her recipes in there, and she won't let it ever leave the house, but she'll let you make a copy of a recipe. And so you make the copy and then go use that. But the original never leaves the house. Well, the DNA strand never leaves the cell, but the RNA can go out and take the information, and then So since we don't want it to accumulate, it breaks apart into its nucleotides. It can be reassembled into another strand of RNA. So the only difference, there's three differences here. The RNA is a single strand. It's a ribose sugar. It doesn't give up its oxygen like DNA does. And it uses uracil in place of thiamine. And that's all. Let's see if this works for us.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide.
Notice on the animation here how the the phosphate and the sugar are bonded together, and the nitrogenous base comes off of the sugar. When I say this, the phosphate and the sugar make up the backbone. They're essentially the outside of the chain of the double helix or the individual strand. So they're running down the outside like of the double helix, or just the middle of the strand for the RNA. It's the nitrogenous base that sticks off to the side in here, and we see even when the chains are linked together, the phosphate is what holds the sugars together, because you'll notice that the phosphate, where it says the P for the phosphate's coming off the sugar here on the ribose sugar, and it will link to the sugar there. So the phosphate links to the sugars on both, above it and below it.
Right. And the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide.
See how it lines up?
RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
So when you see that the double helix strand, let me see if I can get back here.
The alphabet. However, the bases bind around each other to form the
There we go.
thymine.
Now, you can see the double strand here on the left for the DNA. Now, the sequence of the bases dictates the order of the amino acids, and the gene could be on either side. We can have gene, because all the gene is a collection of the instructions. It's just it's a collection of the bases. The order of the bases determines what the amino acids are going to be. And so the amino acids that make up the protein are dictated in that segment. So it's the physical location. We can have a gene on either side of the strand. What's on one side may be completely different from what's on the other side. On the left side, you may have the gene for hemoglobin. On the right side, you may have the gene for hair color or something like that. So but the order is very, very critical to us because that dictates what we're going to make. Okay. Now, this third category here of a nucleic acid type structure is something called ATP. ATP is what we use to deliver energy to the cells. It's a nucleotide based off of RNA, but instead of having one phosphate, it has 3 phosphates. And the third phosphate is held in place by what we call a high energy bond. This is what an ATP molecule looks like. It has adenine and it has its 5-carbon sugar. It doesn't link together with other adenines, with other ATPs. You have the 5-carbon sugar, the ribose sugar, and you have 3 phosphates held together instead of just one. The third phosphate that gets attached here is a high energy bond and can hold about 7.5 kilocalories of energy. Don't worry about that number right now. But we can park it in here. We can park energy in that bond. We can store energy in that bond. And we can deliver that energy to other structures in the cell to do work. In fact, they depend on ATP so that we can do things like contract our muscles and breathe and talk and send nerve impulses that require energy. All of this is coming from the energy that gets released here at that bond. When ATP goes to a site where energy is needed, it will break that bond at the third phosphate, release that energy, and revert back to its low energy. Instead of adenine triphosphate, it'll be diphosphate with what we call the inorganic phosphate. Because when the bond breaks, we don't destroy the unit, we just reestablish the bond. So it rebuilds itself a lot. Now, the only problem with ATP is that it only lasts for 7 seconds. We can make ATP by forcing that third phosphate on there. We do it all the time. We park about 7 kilocalories of energy in there for about 5 to 7 seconds, and then it's gone. And we have to make it again and again. We can't store ATP. We always have to keep making it over and over and over again. That's the only problem with it. So, okay, and that is the end of chapter 2. So, okay. Okay, anybody have any questions about anything? As I said, if you're taking the lab test online, it'll open up at 1 o'clock. If you're taking it in here, I'll be here at 1 o'clock. It shouldn't take very long. It's only 50 questions. So, well, it could have been 100.
Well, that's what I said. She's like, 50 questions. It could have been 100.
That's a good, yeah, it could have been 100. So, but it's all multiple choice too. So, and so if you're taking it online, it will also be 50 questions. It will also be multiple choice. It will be, each test will be completely different because it'll be generated from the test bank. So, and if there are no questions, I'm going to get us out of here, and I'll see some of you in class later on.